will bind. So if you don't have any target, then everything will wash out and you will go to absorbent pad, it will absorb. Okay, so in this case, see, now your label is there, so all the reagent will come, they will react with this label, will form the product, and some redox cycling kind of reaction in the bottom part you can see. So this kind of redox cycling reaction you can employ. Then working on reference and counter, these three part you have to connect with the instrument that is called some portable potentiostat, that is called potentiostat. So in the portable potentiostat you can apply some potential, you can get some like chronoamperometry or chronocoulometry or cyclic voltammetry that you can measure. So if you have more target, then more reaction, and you will get the higher chronoamperometric, higher current, higher charge, or chronocoulometric is how higher charge, chronoamperometric higher current you will get. This is the main very basic fundamental things we are using in this lateral flow assay. Clear?
Yes, sir. Yes, sir. Sir, lateral flow assay, how we use this part?
How we use this part for
Lateral flow assay for any detecting virus or anything.
Yeah, see, this sample contains something like your virus, right? So this will go through this lateral flow assay and it will come and it will bind on this antibody, right? If this virus is specific, and that you can use the secondary some antibody that is also specific with your virus, some antibody, right? So if your virus present, then only it will bind, right? The secondary antibody, this one it will bind, and it will go through this kind of redox cycling reaction, right? This redox cycling reaction will start if antigen present. My antigen means the virus, it can be virus. Clear?
Okay, sir. Okay.
Okay, very simple. And can you explain this washing free? You understand, right? How we are using the washing free method for detection of biomarker, right? Is it clear, students? Hello, students. Is it clear? The washing free principle.
So can you explain one more time, sir?
Say, okay. Let's go back. So washing free principle means it is kind of all-in-one reaction, right? We don't need any kind of washing. So why we need washing, you know already, right? So after primary antibody quotation, you have to immobilization. So you have to add your antigen, your target solution, that you have to wash it, right? So that excess target or excess reagent, excess solution will be removed from the surface. Then secondary antibody, then you have to wash it again. Then only the excess one will be removed from the surface, right? That's why we need some washing to get the higher signal to background ratio. So if you don't wash, then we will get the higher background, right? Is it clear, guys?
Yes, sir.
So why then how we can develop a washing free biosensor? See, so this one is very important for point of care device development, right? So just you can drop the sample, and you can incubate it for like 10 minutes, and you can get the data, right? Something like this. So no need the many step washing. This is called washing free. Just see, this is your electrode. Then you can immobilize the avidin like some kind of linker. You can do any kind of thing, like just for example avidin. Then you can immobilize the primary antibody, like that is biotin related. So biotin avidin, they have strong interaction, so your antibody will be on the surface, right? Now in a tube, see, you can add all the reagent, your solution, all that. I mean, your target means when you add some sample like blood or some serum or some saliva. So this kind of things, that if they that content target, then it will show like this. And on the tube also you can use some secondary antibody, some other reagent, some substrate, some mediator, like all the redox cycling substrate, the chemical that I have shown already. That kind of chemical you can use in a tube, and you can mix it with the sample. Then you can drop it on the sensor surface. See, you can divide it the two part here. Two part. So part 1 and part 2. See, part 1 means if your sample content target, so what will happen? This target will bind on the primary antibody, right? And your solution also contain the secondary antibody that already labeled, like some enzyme. And part 2, it's showing if you don't have the target. I mean, your sample does not contain any kind of biomarker or any disease marker. Your sample is healthy patient case. For the healthy patient case, you don't have any kind of target. So then it will not bind right here. So your secondary antibody, but as you are dropping this sample to the sensor surface, so this sample contains secondary antibody that conjugated with some label. So it will also come to the surface, right? But because of the antigen antibody binding, this secondary antibody level, it will be very close to the surface. You can see because it is attached on the surface. Because of Brownian motion, you can see in the zero case, no target case. So because of Brownian motion, they will be in the, I mean, here and there, because they will be far from the surface. So they will not be very close, far. So we can use this principle, I mean, how far they are. So if they are very far, then we will get the low signal basically. If they are very close, then we will get the higher signal. This is called proximity dependent. What? Proximity dependent, right? So proximity means how close your label on the surface. See, now if you add some chemical, I mean, if you apply some redoxycycline, means you are not adding the separately, because all the chemical you can see in the tube here, substrate is there, mediated there. So they will react, right? Basically they will react with your label, right? So with substrate, they will form some product. And this product will react with some mediator, right? So this reaction will be starting on the surface. And this reaction, if this is the very close, this reaction will be very close to the surface. And very close means you will get higher current. If the reaction is far from the surface, then you will get the lower current. So this principle is called proximity dependent electrochemical reaction. So why electrochemical? Because when you are applying some potential, then this kind of some redox cycling reaction will start, right? Because of the very close, this one will be fast. Because of the far, the zero case, it will be the slow. So this is the basic principle for washing free biosensor. So we can significantly simplify the detection procedure because we don't want many step washing. We can reduce the assay time. Maybe within 10 minutes we can detect. Very less time we need. Clear now?
Yes, sir. Sir,
Okay.
how much distance will be there, like between the surface and secondary antibody in the part 2?
Yeah, see this distance generally we assume. So that I see there is a paper you can see. This paper actually we did in our lab. So we are actually calculated the distance. So if there is a very target, they are very close, and how much distance we can get, that we calculated. So can you search this paper in the supplement information? Otherwise, I have to download that paper. Otherwise, so I can't exactly remember the distance, but there is a calculation I already made. So if you can check this paper, there is already mentioned the distance. Otherwise, I can show you. You just download this paper. You can see the distance. So maybe this one, maybe the x1, and this one the x2. So x1 will be the very, very less than the x2. Clear? Okay.
Yes, sir.
Just check this paper. This is very simple paper. You can read this one also. Okay. Yeah. See, now the whole device here, the washing free, that principle I taught you, that you can basically, that washing free principle now you can basically use it for the biosensor formation. See, this is like a very solid, this is a solid substrate. On the solid side, it can be glass, it can be plastic. On that solid substrate, here working, reference and counter, you can fabricate. So basically you are fabricating 3 electrode system, working, reference and counter, on a plastic substrate or in a glass substrate. So this is can be a gold, this is can be a carbon, so anything. Now on that surface, so this is a working case, so we can immobilize the primary antibody, right, that you are going to detect. So if you are going to detect some virus, so it will be specific for that virus, right? So this antibody. Now on that sensor surface, on the top of the sensor, we can put some paper layer like number 1, number 2, number 3. So while putting some paper, because on that paper we can put all the chemicals that we are using in the solution stage. Here you can see in this, that was in the solution, right? Solution. So you have to make the sample solution and drop it. But in this case, you can see that all are in the dried form, in the solid stage. All are in the dried form. Means here, see, ruthenium hexamine or methylene blue or secondary antibody. So these all the chemicals you can keep in the dry form. So they all are on the paper. See. Now on the top of this sensor, we can put a hole. On there, we can put this like blood sample, right? You are dropping some blood sample. Also, if you think that blood sample is very low, like very small, you can drop some PBS also, or some buffer also with the blood sample. So it will help some buffer to pass through all the membrane. So basically, you can add your sample, your that you are going to test. Then see the here in this membrane. Here, this is also kind of a membrane. See, all are the paper based. So this is also membrane. This is called vivid plasma, PLSMA, vivid plasma separation membrane. So one paper we can use. So this paper will help you to separate all the blood cells. So all the blood cell will be here, and your plasma, means the liquid part, will go through this all the membrane. So what will happen? All the reagent will dissolve. Then it will come to the actual sensor surface. Now your target will bind on the primary antibody that already quoted, and all the reagent will come. And when you will apply some potential here, like 0.3 volt, so your redox cycling reaction will start. Then you will get the like chronocoulometry or chronoamperometric data. And like this way, you can measure the unknown concentration. You can make calibration curve, everything. So this is kind of the washing free sensor you can develop. So no need many step washing, just drop, and you have to wait like 10 minutes, 10 to 15 minutes you can wait. Why we need some time? Because you should go through all the paper, then it should come to the surface, then it will bind, or the reaction will happen. So for that we need some time. That is called incubation time. That you need. Then you will get the result. So this is very, very simple biosensor, and it will be useful for point of care. So now you can ask, because this kind of the antibody, this kind of thing, they can be denatured at the high temperature, right? So how it will be useful for point of care? So for that, you can drop some stabilizer, some solution we have, or instead of antibody, you can use some aptamer or some DNA base. And that will not be denatured easily at the high temperature. So this kind of technique, if you use, basically this will be very useful for point of care diagnosis. So you can think about. So this is just a very generic sensor. You can develop similar kind of biosensor for any kind of disease detection. Okay, guys, please ask any questions if you have. This is very simple. Is it clear?
Sir, do we remove the all papers after the test is completed?
No. So it is kind of very simple chip like this. So all the paper will be the step by step, always stacked on the surface. Will not remove, just you will drop, and this part will be connected to your potentiostat, small potentiostat. Then just drop and use. So you will get the data. So after use, you can throw this chip. So actually this kind of single will use and throw. Okay.
Okay, sir.
So any more questions? It's very interesting biosensor, some device. It is full complete lab on a chip. So what is lab on a chip? That I already taught you. So lab on a chip. I mean, so on the chip, all your lab reaction, right? Like the chemical reaction, your attachment of the antigen on the surface, signal amplification, that kind of all the steps that you are doing separately in the lab, that everything can be done on a chip at the same time. Then that's why it is called lab on a chip. Clear, guys? Can I go to next topic? Or if you have any question, please ask me.
Sir, hello, sir.
Tell me.
Sir, what is the ABCDE, the part? Please, sir.
Oh, this A means just part by part I mentioned, because A means the sample drop. This is the blood. B means your vivid plasma separation membrane that will separate your blood. C means some chemicals will be here, like some secondary antibody or some methylene blue secondary antibody I put in the D part. Like here you can put some methylene blue. Second, here also you can put some secondary antibody. E part you can put some ruthenium complex. I mean, it is not strict. Like you can use any kind of reagent on the one by one the paper. So this is kind of F means your sensor surface, so something like this. So we just denoted A, B, C, D, like so many layers here. So actually they will be on the stack one by one. We will put on the sensor chip. Clear?
Sir, in the below, what is it is label antibody?
Yeah, so in the below you can see this one is the primary antibody on the surface. So this is your
Oh, okay.
that I told you. This is your sensor, your biosensor. All it can be the primary antibody. It can be the aptamer. It can be any DNA. Okay.
Okay, yes, sir. Then the work of REWOC, this pattern, please, sir, you told me.
Yeah. So you can see. So here actually I told you I fabricated just 3 electrode on the surface. Like W means working electrode, R means reference electrode, C means counter electrode. Right?
Yes.
Clear?
Yes, sir. After sample drop and it is go to the below, then how it is working?
So after when you drop the sample, so here is drop the sample, right? A.
Yes.
So when you drop the sample, see here B is a vivid plasma separation. I mean, it will separate all the blood cell. Then you will get the liquid part, right, from the blood. That is plasma. Then it will go through the your all the membrane. So it will dissolve all the chemicals. Also, if your sample contain antigen, I mean target, then it will come to the surface. Then it will bind on the primary antibody. Okay. Okay. Clear.
Okay, sir.
Okay, so in this paper here, actually I mentioned one paper. You can try to download this one. You can see that low interference was increased. So one sensor actually we developed, that is troponin I, one heart disease biomarker we can detect like this way. So you can read this paper basically that I taught you the all the principle that we described in this paper. Here, as I mentioned last ascorbate oxidase why you are using, because our real sample like blood contain lots of ascorbic acid. So you can see the right side the CV, this is cyclic voltammetry of the ascorbic acid. So see, you can see the lots of contribution, I mean lots of current from the ascorbic acid. So if you apply like 0.3 volt potential, so you can see the ascorbic acid can be oxidized, right guys? It can be oxidized on this potential. Clear guys? From the CV, it implies that it can be oxidized, right? This red one.
Yes.
Now, if we add the ascorbate oxidase, that 5 unit per ml, some concentration, if you add, you can see this one will go down like this blue one. It means if you add ascorbic oxidase, so this effect will be removed. So you will not get any contribution. Means your background will not be affected by the ascorbic acid. So that's why we use such kind of the chemical. So when we develop some wash-free biosensor, we'll try to eliminate the interference species effect, right? So interference species, there is lots of interference species in the real sample. So as it is not washing, it is wash-free sensor, right? So all the interference species will be there. So we have to avoid. Like say here UA, mean uric acid. You can see the uric acid is oxidizing around here in high potential, but you are applying like 0.3 volt. So it is fine. No need to eliminate this effect. Right? Clear guys? See uric acid oxidizing here in this potential, but you are applying here. So in this case, no need to eliminate this effect because you are applying lower potential. Clear? Clear, guys? Hello?
Sir, we know it's oxidizing because the current is increasing, right?
Yeah. You're oxidizing because current is increasing, right? Because see, it's going like this in the background. Then it's oxidizing. Oxidizing means because current is increasing. So you have to, otherwise you will get this current in your background. Clear?
Okay, so if you use this, if ascorbic acid or any, it's present on the sensor, so at that potential the background will be increased. Am I right, sir?
Yeah, you are right. So your background will be increased. So if you apply like 0.8 volt, see, you will get high background, right? Because this much oxidation you will get. But if you apply 0.3, we almost this is almost the zero, no current, right? You will get the lower background basically. Clear? So this is the main things. Means in the washing free biosensor, so you have to avoid this kind of interfering species effect. So one main part or main technique you can use, the application of potential. I mean, you cannot apply very high potential in the washing free biosensor. So you have to apply around here, because you will see in this case, you are almost no current getting, right? So in this, in the ascorbic acid case, the cyclic voltammetry, it is oxidation, you start from here. So almost this potential no current, almost this potential no current. There is another interference species like acetaminophen. You can see that here almost no current. It is oxidizing higher potential. So don't use the very high potential in the washing free biosensor. That's why. So this kind of biosensor case, generally we are applying the applied potential. That's why around 0.05, very low potential generally we are using. So you have to optimize which potential is the better, right? So you can find out like 1, 2, 3, 4, 5, like so many potential you can apply, then try to calculate signal to background ratio and calculate the best signal to background ratio which applied potential. Clear? Guys, is it clear now? Washing free sensor
Yes, sir.
technique. It's very interesting. Means this is the technique that I am teaching you, that you can use for your actual point of care sensor development. This is very innovative technique. Okay. This one, is it clear, right, for you? Aman Chaurasia. Hi, Aman. Are you there, Aman?
Yes, sir, I'm there.
So how is it? In this sensor technique, is it okay? You understand this one?
Yes, I'm trying to understand it.
So if you're not clear, you can ask me. I can repeat. So which part is
Yeah,
not
yeah,
clear?
sure, sir.
Which
Sure,
part is?
sir.
So try to understand this one. So you can ask me now only, even if you're not clear. I am just repeating again and again. So like this way, actually, we can develop very good sensor.
Yes, sir.
See, so we just obtain the potential. Now you can see. So we can now calculate the different concentration. This is the chronokilogram, right? Different concentration, charge versus time. Now we can make some calibration curve. Now from this you can detect the unknown concentration, right? So this is that's the same thing, same technique you can apply for any kind of sensor. Okay, clear? Let's go to the new topic then.
Yes, sir.
Yes, sir.
Yeah, now I will show you, I will teach you one more thing, the optimization technique that I taught you, right? Different potential we can select, then we have to calculate signal to background ratio. See, so background means, as I mentioned, that ruthenium hexamine GP. GP means the substrate. I mean, in this reaction, you can see what is the background, what is the signal. Can you tell me in this reaction? Like GP means some substrate, your GPDH means some level, your ruthenium means ruthenium hexamine, your mediator, right? So if you have target, then it is the signal, right, guys? If you have target, then it is signal, right?
Yes, sir. Ruthenium, that is signal. Yeah.
And if you don't have target, then it is background. I mean, see, if you have target, then GPDH will be on the close to the surface or immobilized on the surface, right? So then you have to think about that signal means GP, GPDH, and ruthenium. All the chemicals will be involved, right? And background means maybe GPDH will not be there, or they will not will be close to the surface. So basically, GP and ruthenium, they will take part in the background, right? So if there is no target, maybe GPDH will not be on the surface. Clear? So GP, ruthenium will be the background current they will generate, and GP, GPDH, and ruthenium, they will generate the signal current, right? Right, guys? Like the redox cycling reaction I taught you last time. This is actually enzymatic redox cycling. Clear? Can I repeat, or is it clear? Clear, right? Okay. Now you can see. So ruthenium GP, we think this one is the background B. And ruthenium, hexamine, GP, we think about this is the signal. Now see, we choose different potential like minus 0.05, 0, like then plus 0.05, then plus 0.1 volt. So many different potential we choose. And when we compare signal to background ratio, then we saw that 0.05 here actually we are getting the highest signal to background ratio than other applied potential. Okay, so that's why we optimize that 0.05 volt is the best one for this biosensor. So for this biosensor, so 0.05 volt is the best one. So this way we can optimize the applied potential. So in the washing free biosensor, applied potential is very crucial part, very important part. Clear? Students, is it clear?
Yes, sir.
Good. Now I'll teach you some another technique. See, so this is called something like the aptamer. Aptamer means it's nothing but it is kind of the sequence, DNA sequence. You can see ATGC, and EP means this is the electroactive probe, like the label. So you can immobilize your one kind of this. This is actual. It is kind of the receptor. Some all the ATG sequences here. Like you can say this is the DNA, or this is the artificial one where we can synthesize. This is called aptamer. Okay. So this one at one end we can conjugate one EP. EP means electroactive probe. So what is the electroactive probe? Electroactive probe means like the ruthenium or like the methane blue that I taught you, right? So they will generate the signal. They will generate signal. Okay. See, if this probe is far from the surface, then the electron transfer from this EP to the surface will be slow. And if this EP is very close to the surface, then the electron transfer from this electroactive probe to the surface will be fast, because they are close. This is also proximity dependent. This is far. So if you have some target like toxin here is one of your target. So if your target bind to this aptamer, so they will fold and they will come close to the surface, this electroactive probe. Then you will get the high signal. So basically you can see this is the higher signal and this is the lower signal for the no target case. If there is no target, you will get lower signal, and if you have the target, you will higher signal. Why higher? Because electroactive probe will come close to the surface. Guys, clear now? Hello? Hello everyone.
So why it is getting folded up?
Oh, so when actually this is kind of the synthesis, we're using different kind of technique in the aptamer industry. They're trying. This is kind of they're calculating the binding energy and their behavior. So it is kind of like toxins or any kind of target when they bind. So it can be folded, it cannot be folded. But aptamer chemistry is something the industry people they can try, and that technique actually we are not teaching here. So this is basically when they bind here. So there is some energy change, the binding energy change, some conformational change, mainly the conformation, mainly conformation change because the binding energy change. So they will come close to the surface. So this is some different scores that I'm not teaching here, but just I'm showing you some conformation will change. Because of that, this will be very close to the surface. Okay, clear?
Yes, sir.
This is some conformation change, so you will get it. So this is very simple, like a washing free sensor, right? So you are not going to use so many chemicals, right? Just drop the sample and you can wait for a few minutes and you will get the current. So this is very simple technique you can use for device development basically. So basically device development you can use this one. Okay, good. Now I can show you this on some level free wire sensor. So I taught you the washing free level. Now I am going to teach you the level free. So what is the level free? Level free means your secondary antibody will not be conjugated with any label or any electroactive probe, right? Like enzyme or any methane blue. No. So if it is not conjugated with any label, that is called level free sensor. So for the level free sensor, generally we are using the impedance, electrochemical impedance spectra. I think I already taught you this one, S mutant detection. Like primary antibody will be on the surface. Then you can block the surface with BSA. Then if you have the target like S mutant on the surface, then it will bind, right? So what will happen? The electron transfer rate from number 1 to number 2, then number 3 and number 4. You can see the electron transfer rate is slowly. What will happen? Electron transfer rate slowly increase or decrease? Guys?
Decrease.
Yes. So electron transfer rate slowly decrease. So you can measure that one. So that is called the electrochemical impedance spectra. You can measure the impedance basically. So you can measure the RCT value. So generally in the level free wire sensor case, you can use this impedance spectra or RCT measurement, and you can plot like different concentration. You will get higher and higher RCT, and you can plot it in a calibration curve. Then you can get an unknown concentration you can measure. So this is called level free sensor. Why? Because you are not using here secondary antibody that is conjugated with some label, right? That's why it is called level free sensor. Clear, guys?
Yes, sir.
Yeah. Now I am going to teach you some new things that is called multiplex sensor. But before that, I will come to you. So I already taught so many topics. We will have exam. Maybe I will announce you the exam. Let's ask you. Maybe as you have the already Viva on Wednesday, let's reschedule it on maybe Saturday. So what about you? Are you free Saturday sometime around morning, 10 to 11? Is it okay, guys? Can we have one class test Saturday 10 to 11?
Sir, not possible. So Saturday I have exam on that day.
You have exam. Okay. What about others?
So at 10 to 11 I'm having exam. So.
What about the then afternoon session around 3 to 4 or 4 to 5? Is it okay for all of you on exam Saturday?
We have practical class, sir. MTech.
Okay. Yeah,
We
this
have
is,
a class.
this is the difficulty thing because you are so many different. Means you are here from the other departments. So
Sir, can we directly have it on the Monday class and next Monday class?
Yeah.
okay.
Yes,
So
sir. Monday will be better, sir.
Okay, let's continue the class then whole week, Wednesday also. Then Monday we will have the class test. Okay.
Yes, sir.
Okay, sir.
Okay, sir. Okay.
I will send the email. So Monday we'll have one class test. And please read until then Wednesday class. Then we will include the some MCQ type questions. So up to Wednesday's class, you can from the first class to Wednesday's class. So you can read and you can try for your test. I think very simple technique that I taught you. That very innovative technique. If you understand, you can answer all the questions, right? What about Abhijit Desmukh? Abhijit, how are you, Abhijit? Hello, Abhijit, are you there? On hold. Akash Sharma. Hello, Akash. Hello, Akash. What do you think? I mean, do you have any kind of, if you have any questions, if something is not clear, do you want to discuss something? Do I want to repeat? Can I repeat something? So everything is clear to you, Akash? I think your voice not coming. Okay.
Hello, sir.
Akash, can you hear me?
Not Akash, sir. Suman.
What happened?
I have.
I think some problem. Okay. Anuprabha Chatterjee? Anuprabha. Hello, Anuprabha Chatterjee. How are you, Anuprabha? Are you there, Anuprabha? No? Hello? Jais Sharma? Hi, Jais.
Yes, sir.
Okay. So everything clear, Jais?
Yes, sir. Sir.
Okay. So I think now you can think about some new plan, something that you can read some papers. I will recommend all of you, please search some paper in Google and just try to read some paper, some new technique that I already taught you so many things. You will get the basic idea. I think you already got the basic idea. So now you can understand all kind of the bioscience they develop already. That's why I'm going to schedule your presentation maybe at the beginning of the next month. So it will be good for you. Okay, and what about the Nilesh Rathod? Nilesh Rathod. Hello, Nilesh. Are you there?
Yes, sir. Hello.
Okay. Is it clear everything? All the topic that I'm teaching you?
Yes, sir.
Okay. So what is your background, Nilesh? You are from mechanical, right? No.
Yes, sir. Mechanical, sir.
Okay. So are you do you think is it interesting for you? Means you can use your mechanical background for development some device here.
Yes, sir. It is interesting, sir.
Hmm. Means this is not kind of simple course. So this is kind of the practical, right? Very practical oriented. So while you will be here, actually, I will show you some experiment in my lab. But just think about so how you can use your knowledge, your idea for development of the sensor. This is kind of the very practical knowledge you are getting. A very good course you are learning actually. Okay, okay, Nilesh. Okay,
Yes,
Nilesh.
sir. Yes, sir. Okay, sir.
Okay. Good. And what about Anmol? Anmol?
Yes, sir.
Okay. So everything clear to you?
Yes, sir.
Okay. So
Sir, one clarification, sir. Hello.
okay, this one. Tell me.
Sir, you wrote a chapter on this book, immunosensors book, right? Like
Right.
the redox cycling technologies for point of care. Sir, is
Yeah.
it enough for this signal amplification part? That chapter is enough, right? Is that enough?
Yeah. In that chapter, actually, I summarize everything. I summarize. So it will be enough actually. But I will recommend you just read the chapter. Then just you can search some technique that is available. I mean, some group they're modifying, right? So many groups are there. They're modifying this kind of amplification. I mean, you will get
Yes, sir.
all the basic idea that I can assure you. Okay.
Okay, sir. It's been 1 month, so we have to revise the topic that signal amplification. So I was thinking to read that chapter.
Yeah. You just read that chapter. This is very good and interesting chapter. You will get all the ideas. Then you can
Okay.
read some other papers. Okay. Yeah. So I hope you will be here by May, June, something like this. So if you have any time, please visit my lab. I will show you some practical something, practical some experiment. I will show you how the signal actually amplify by using the redox cycling. How you can fabricate on the device. I have so many device design. I have some biosensor platform that I will show you. So okay. Okay. And Bhavesh Datla, how are you, Bhavesh? Hello, Bhavesh. Are you there?
Hello, good evening, sir.
Bhavesh, good evening.
Hello.
Bhavesh, good
Yes, sir.
evening.
I'm good.
So, is it clear? Everything?
Yes, sir.
So, how much you got
Yes, sir.
in the last test?
Sir, I got 19 out of 20.
Oh, really? Very good. Means you understand everything. Nice. So, then whatever I'm teaching you, so please try to. So you are from a mechanical also, no? No, not electrical. Maybe I forgot.
No, sir. I am from food engineering.
Oh, you're from food engineering. Okay. So see, now in the food engineering case, you can try to means why I am asking name by name in your background, because just use your background. So I mean, so you can design some sensor for detection of some food pathogen, right? But quality of the food, right? Hmm. Right, Bhavesh?
Yes, sir.
Hmm.
Yes, sir.
Also, you can detect some soil quality, right? So you are cultivating some food. I mean, you should know the quality of the soil, right? Maybe there is some pesticide, some other harmful chemical, maybe arsenic there, or in the water, maybe from the soil, but from your food, maybe some contaminant are coming to the drinking water, right? They're contaminating. So that also you should detect. Clear, Bhavesh?
Yes, sir.
Hmm. Yeah. Yeah, like this way.
Hello, sir.
I am motivating actually you. Yeah, so you just use your background and try to develop something from your area. Hmm. And then I will be very happy that I taught this class and you are benefited actually. Okay. Tell me.
Sir, is the which thing you upload in file session that is enough for study, sir?
Yeah, that is enough for your fundamental knowledge. That's why I'm telling you. You should search some papers. I can help you. I will download some papers and I will upload you by tomorrow. Okay. Some
Yes,
paper.
yes, it is very helpful, sir. It will.
Okay. Okay. So I will work for you then.
Oh, okay, sir. Thank you, sir. Thank you.
But I will recommend you work some. You should also work hard. Okay, I'm also
Oh,
working. I'm working hard
of
for
course.
you. I'm working hard for you so that you will get some knowledge. But you also
Okay.
try to find out something. See, while you will search, see one thing. I will show you. Just go to Google. I'm sharing my screen. Okay, then search electrochemical biosensor for cancer detection. Okay, can you see?
Yes, sir.
So just click it. Okay, then go to image here. Third one, image. You can see there is so many sensor. Just try to find out which one you can feel that yeah, it is very interesting. I want to read. Right? There is so many sensor image you can see. Then you thought, oh, it's really good one. Good picture is showing some antibody they use, some amplification they use. Okay, let's click this one. I want to read it. Then you can see the micro machine free full text this paper. Then click it. Then if you click it, then you will get the paper. This is the electrochemical biosensor for determination of the this kind of the some direction. Then download the PDF. Then you can download this, and some paper has a free access you will get, and you should contact the IT the library, and you will get some access if you are not able to download some papers. And you can accept the cookies, and then your download will be started. Then you will get the PDF file. Okay, is it clear? Is it interesting, right, Suman and others?
Yes, yes, sir.
No. So like this, so you can search so many papers. See, I already taught you all the basic things. Actually, I taught you. Now, if you go some paper, something, if you search, you will get many idea. Okay. Okay, let's go back now.
Okay.
Now let's go back some multiplex detection. So until now, I taught you the single detection, right? Single means only one analyte you can detect, and multiple means so you can detect at the same time like 2 marker or 3 marker. So what you will do? So maybe like this is kind of paper strip, right? So you can maybe one working one and another working two. So here you can immobilize like suppose this is the cancer, and suppose here maybe you are going to develop some like malaria detection. So one antibody you can immobilize on working two, and working one you can immobilize maybe some cancer, right? Or you can increase number of like working one, working two, working three, working four. So many working electrode you can fabricate. Right? Instead of using one, you can actually try array. So many at the same time. So I will show you here one multiplex sensor. If you see, this is the actual sensor design here. So here is the working 1 and here is the working 2. The working 1, actually you can immobilize some antibody here, and working 2 you can immobilize another antibody. So it should be specific for 2 disease biomarker. So multiplex means you are going to detect more than one, right guys? Right?
Yes, sir.
So you're going to do more than one. So you can use the wash-free now technique. Like you can drop the blood, your all the membrane will be top of the sensor, your reagent will be here. But instead of using one antibody, you can try another antibody. So you will get then two signal. In the working 1 case, you will get one signal. Working 2 case, you will get another signal. Right? So in this case, working 1 case, you will get one calibration curve, and working 2 case, you will get the another calibration curve. So basically, on a portable electrochemical analyzer, on the display board, you will see the two curve. One is for working 1, another is for working 2. Then you will get the concentration of the two biomarker on your screen, right? So this is a very simple technique. Like instead of using the 1, you can use 2. Also, you can try to increase number of the like working area. Means you can fabricate. So basically, we are fabricating or something design. You can design, like here actually the mechanical guys, you can use your knowledge. Electrical guys, you can use some your electrical component. I mean, how you can connect all the electrodes, how you can fabricate your all the electrodes on the, like you can use some plastic substrate, you can use some paper substrate. So something like very cheap material you can try, not the gold, something like this. See, here working 1, working 2, working 3, working 4. So 4 antigen you can detect, right, at the same time. So that's here actually see, you can see some PBS we are using after dropping the blood. So why? Because maybe you drop when you're pricking your finger, maybe you will not have the enough volume of the blood. But if you drop some PBS, then it will help you to pass through all the membrane. So you can use some buffer solution. So your final chip will be looks like this. Clear? So your final chip will be looks like this. So you will drop, then it will go through all the membrane, and it will come to the surface, and reaction will start. Then you can connect these things with a portable potentiostat.
Sir,
Yeah, tell me.
sir, actually, I am asking that we are having here 4 targets, so we are having 4 signal. Then won't this make our sensor a bit less sensitive, or won't this thing interfere with, I mean to say, how biomarker will be detected simultaneously? I am not getting this thing.
No, it will not affect basically. See, here you can see the 4 actually we measure. Like this is kind of the like cycling voltmeter on the oxidation part, like working 1, working 2. See, here actually you are eliminating by the eliminating all the interference effect by using some ascorbate oxidase, by applying some lower potential. So this kind of thing you are actually eliminating. Okay. Now, when you maybe your sample contents generally see lots of biomarker present, right? Not only cancer, maybe dengue, malaria, tuberculosis, so many things. But if you have the antibody on the surface, then only they will bind. Otherwise, they will not bind, right? And if they bind, then only you will get the signal. So basically, it is very much optimized system. It is I can't say that it is very easy, but it is very much optimized. So you have to optimize which potency you are going to use. So how you are going to fabricate this working electrode, counter electrode, reference electrode, and also, I mean, how much volume you need, how much reagent you need. So everything you have to optimize so that you can minimize the background and you will get the higher signal. Okay, clear?
Yes, sir.
It is fully optimized system, but it is not very simple task. But after optimization, it will be very simple device, that I can assure you. Okay. So that's why generally for development of this kind of devices takes some time. It is 3 years almost for one sensor. Generally when I think some of you already know how we are running the project. Generally, this kind of project we are running for 3 years. Why? Because first 1 year actually we are trying to solve this kind of interference cases. Then second year we are trying to develop we are trying to fabricate this device. Then we are trying to integrate this sensor with some electronic component. These things slowly we are overcoming the problems. Okay. It is not like overnight work. Okay. Okay, clear?
Yes, sir.
But it is very you need some hard work, and you can, once you optimize, your sensor will be in your hand. You can easily commercialize. Yeah, so this kind of things you can see one report, some multiplex sensor, like very simple. Instead of using antibody, you can use some enzyme, like for glucose oxidase, lactate oxidase, uric acid, some their oxidase we can use. So if you drop your sample, so glucose can be oxidized here, lactate can be oxidized here, uric acid can be oxidized. So you will get the label like glucose, lactate, and uric acid in your sample. So at the same time, you will get the 3 concentration label. Clear? So this is kind of some fabrications of the biosensor. See, on a paper, as they fabricate, like this is a carbon-based material. They fabricate on the paper, and then just very simple. This is a very simple device. Then you can drop the sample, and you can connect this one with your potentiostat. You can connect, and you will get the data. Fine.
Yes.
Do you have any questions? Guys, do you have any questions? No? It's very simple, right? Yeah, very good. So, yeah. So I taught you actually so many new topic like watching field sensor, and like it's a device multiplex sensor, so many things. I covered the device design. I covered the redox cycling. I covered the lateral flow assay. So many different kind of example of the redox cycling. Right. Then how the amplification work. Why we need less sensitive electrodes, chemical amplifications. Right. So this is a vast area actually. I'm just covering. So please, I will recommend you please read some papers, actual papers. So I'm planning tomorrow, as I promised. I will try my best to upload some papers. I will download, but I already taught you how to search the papers in Google, right? You can search also, but I will try my best to upload some papers also for it. Okay, guys, do you have any questions? These things, if you want to repeat, everything fine? Bhupendra?
Yes, sir. Sir, need to study first. Then, if any question is there, then in the next class I will ask.
Yeah. So that's what I recommending. So please, just this is not kind of thing you are attending the class and you are not reading in your home. So please read, because I am expecting more questions from you. I will try to my best to answer you. And from my expertise, I think I will try everything to clarify, so everything should be clear to you. And I will be happy if you can develop something by yourself. It should not be like this, like a course. Not on. I mean, it is not like the coursework. It is kind of knowledge you are getting, so that you can use your expertise. Clear?
Sir, I want to say something to you.
Yeah, Bhupendra, sure.
Sir, actually, when you said you have to read the paper, to be very
Yes.
honest, sir,
Yes.
reading paper is very rigorous task. So,
Yes.
I want to ask that, is there any way that we can enhance the reading ability of reading papers or something like that?
Very
Because
good question. Bhupendra, very good question. So why not then? One thing I will tell you: please download some papers and face the problem. Why you are not getting the idea? Next class, I will teach you how to read the paper. Is it fine?
Yes, sir. Yes, sir. Because this subject is more like of inclined towards that thing. Because in
Right.
yes, sir.
Right. So I will recommend everyone, please download some papers, try to understand, and tell me next class why you are not getting the idea. Right. So I can teach you today also, but I'm giving you one day time. Please download some papers and try to understand that technique, and tell me why you are not getting, because you will say definitely some problems, right? Because of this is not clear to me. I don't have this background, these things. Then I will teach you how you can read